Hello, I'm here today to tell you a story about how studying fundamental properties of gene regulation can help enrich our understanding of how life begins during development and how diseases such as cancer can manifest in instances of gene dysregulation. And before I get to the heart of my story, I'll briefly explain how I got here. You see, in the scientific community, I'm what you would call a basic biologist. No, that doesn't mean that I walk around the lab in Ugg boots taking selfies while drinking a pumpkin spice latte, though I do love me some avocado toast. Being a basic biologist means that I take complex biological questions and distill them down to their fundamental properties to provide insight into the molecular and genetic interactions that lead to a particular biological outcome. Being a basic biologist means I get to satisfy my neurotic and anxious mind by answering all of my burning questions, like who, what, where, why, and how. So let's start with who. Who is this story about? Well, it's about you, and me, and them, and all of us. We are complex creatures full of mystery and wonder and questions that need answering. Now, what brings me to this stage? Well, if you reach back in your memory, back to your high school biology class, you might remember learning about the central dogma of biology. And that is this: all living organisms have genetic material called DNA, and within that DNA are our blueprint of genes that uniquely define who and what we are. Those blueprints are read off as messages made up of messenger RNA or mRNA. And these temporary messages are then encoded into the final product, protein, which carry out the enzymatic reactions that occur in our cells. Now, as you might imagine, each of these steps requires a checks and balances system to make sure the cell has just what it needs to function properly. You could picture this as an assembly line. Without any external perturbations, say from a supervisor, the assembly workers transcribe the DNA into RNA and subsequently translate the protein with inspections by quality control at each step to maintain a normal level of activity. When there's a change in the environment or cellular status, say in the case of an infection, for example, that requires a dramatic increase in activity, the supervisor tells the assembly workers to make more mRNA and protein while the whole system ramps up to meet the needs of the cell. When those needs are met, the assembly line slows down and the cleanup crew comes in to discard all the excess mRNA and protein to bring the activity level back down to normal. In reality, all of these key players, the supervisor, the assembly workers, the quality control, and the cleanup crew, are regulatory proteins that each play a crucial role in maintaining this well-oiled machine. My research project aims to understand what regulatory proteins regulate my genes of interest, how they do their job, and what happens when things go wrong. More specifically, I study the regulation of genes whose function is important for a critical patterning process in development of all vertebrate species. These genes have a very interesting expression pattern in which the levels of mRNA and protein fluctuate in kind of a wave-like manner as shown in this diagram. We describe these genes as having an oscillating expression pattern, and this is actually considered one key hallmark of genes involved in stem cell maintenance, as is the case with the genes that I study. As is typical in biology, in order to understand or identify the regulatory proteins that keep these oscillations going, we need to study this in the context of a broken system. For example, what happens when these temporary messages aren't so temporary? When the mRNA cleanup crew is on strike. Previously, my lab has demonstrated that a loss of function of a regulatory protein that helps degrade mRNA results in stabilization of our oscillating genes. Now, as you might imagine, without the mRNA cleanup crew, protein would continue to be made at the wrong time and gene oscillations would be entirely disrupted. But fortunately, nature is cleverer than our imagination. What we found is that although the mRNA is present at a high level when it shouldn't be, the protein continues to oscillate and developmental patterning is unchanged. We now have evidence that suggests that regulatory proteins within our quality control directly bind the mRNA and prevent the translation machinery from translating too much protein. We are now currently working to identify which specific RNA binding proteins are responsible and determine whether altering their activity results in defects in mRNA degradation and translational regulation, resulting in developmental patterning defects. By now, you may be thinking, sure, development is cool, but what about after we develop, are born, and grow up as fully functioning adults? How might these regulatory programs impact our lives today? Well, in fact, some of the same RNA binding proteins that I study have several implications in development and disease. More specifically, one of those classes of proteins called AU-rich element binding proteins has a very diverse set of functions and associations with cancer. And in some cases, the function of one protein can directly oppose the function of another. For instance, the protein HuR is commonly elevated in several malignant cancers such as brain, breast, cervical, and lung cancer. And this is likely due to its role in enhancing cancer cell proliferation, facilitating evasion of the immune system, and promoting cancer cell invasion. And it does so by promoting the stability and translation of cancer-associated mRNAs, essentially providing the cancer cells the fuel they need to survive. In contrast, another protein called TIA1 is commonly downregulated in multiple cancer types. Previous studies have shown that in a healthy cell, TIA1 modulates the expression of mRNAs that promote cell death and arrest cell growth. However, when downregulated, this leads to uncontrolled cell growth and reduced cell death, causing cancer. The more we can understand the context-dependent nature of these types of regulatory mechanisms and how these programs are utilized by cancer cells, the better we can approach therapies that modulate RNA binding protein activity. Using this strategy, we can target specific mRNAs to modulate expression and restore balance in our cells. And perhaps then we can take back control over these mechanisms and stop cancer in its tracks. Thank you.